Okay, Assalamualaikum warahmatullahi ta'ala wabarakatuh. Okay guys, so today we are going to continue with the topic of movement of a substance across plasma membrane, part 2. Okay, so I hope you guys still remember what we have learned about uh, the movement of substance across plasma membrane, part 1. Okay, so this is the next thing that you should know. Okay, so let's move on. Okay, so previously we have studied about there are two types of transport. Okay, the first one is passive transport. Okay, and the next one is active transport. Okay, so I just want to give you some revision about what is the difference between these two transport. Okay, so basically passive transport, okay, is a transport of substance without using any energy. Okay, without using any energy. Okay, but for active transport, okay, it involves energy. So that's why it is active. Okay, so these are the example of passive transport that actually happen in our daily life. Okay, so we go one by one. The first one is passive transport. Okay, occur in organism. Okay, during the first one, okay, gases exchange between alveolus and blood capillary through simple diffusion. So as you can see here, this is one of the example of a passive transport. Specifically, this process is called simple diffusion, where there is a gaseous exchange in the alveolus and blood capillary, where the oxygen from alveolus, okay, will diffuse from alveolus to blood capillary, while the carbon dioxide will diffuse from blood capillary to the alveolus. Okay, and this process is called simple diffusion. Okay, and the next one, reabsorption of water, okay, occur by osmosis in the renal tubule in the kidney. Okay, renal tubule is a name of cell in the kidney. Okay, so the process is reabsorption of water. Okay, and it is done by osmosis. Okay, and then the absorption of water by plant root hair, okay, by osmosis. Okay, and the last one, absorption of fructose molecule, okay, in the villus by facilitated diffusion. Okay, for those who still remember, facilitated diffusion, usually we will use, okay, channel protein. Okay, channel protein yang ada lubang tu. Okay, yang the substance can just easily go in and go out. Okay, so here I would say passive transport. Okay, ni satu lagi ni, awak tengok. The absorption of water by osmosis. So root ni dia akan absorb water. Okay, sama macam ni lah. Okay, absorption of water by plant root hair cell. Okay, by osmosis. So if in the exam they ask you, please name the example of passive transport. So this is could be your possible answer. Okay, the next one is the example for active transport. Okay, this happened in your daily life. The first one is absorption of glucose and amino acid in the villus. Okay, villus is in the small intestine. Okay, so when we want to take oxygen and amino acid, okay, into the small intestine, we require energy, and that process is called active transport. Okay, reabsorption of glucose through renal tubule in the kidney. Okay, when the kidney absorb water, okay, it is called osmosis and passive transport. But when the kidney absorb glucose, okay, it use active transport. So you can see the comparison. And then the transport of sucrose from leaf to phloem tissue. Okay, so just to remember, phloem is a plant tissue that will transport nutrient. Okay, the nutrient will also include sucrose. Okay, so the transportation of sucrose is called active transport. And the last one, absorption of mineral ion by plant root hair cell. So you see here, root ni, it will absorb two things, water. If it absorb water, it is osmosis and passive transport. But if the root absorb mineral, ni awak tengok mineral salt, okay, mineral ion, okay, and it will use active transport. Mineral ion tu ambil pada mana cikgu ni? Mineral ion basically it take from fertilizer, baja. Okay. Okay guys, so that's all for this one. Let's move on. Okay, where is it? Okay, here. So the next one, we are going to study about the types of solution. Solution dalam bahasa Melayu, larutan. Okay, so you say that there are three types of solution. The first one is isotonic, and then we have hypotonic, and the last one, hypertonic. Okay, so we go one by one. Okay, so okay, I'll tengok sini ya. So basically, the diffusion of water will occur by osmosis. Okay, right now for this topic, you have to focus on osmosis. Okay, we want to talk about osmosis because this topic basically will involve the movement of water. Okay, the diffusion of water. So when it is the diffusion of water, it means that the process is called osmosis. So for those who don't remember, osmosis is a process where the water diffuse, okay, from an area of higher water potential to area of a low water potential across plasma membrane. Okay, maybe some of you might be wondering, teacher, what does water potential mean? Okay, water potential and water concentration. Okay, they deliver the same thing, the same meaning. Maksudnya, banyaklah kepekatan air tu. Water potential dengan water concentration are the same thing. But I prefer you to use the term water potential. Okey, maksudnya contohlah air kosong kan. Air kosong ni dia tak ada apa-apa, dia ada hanya air saja. So I would say air kosong tu has higher water concentration. Ataupun cikgu boleh cakap air kosong yang tak ada apa-apa tu, it has higher water potential. Okay, water concentration, water potential are the same thing. Okay, boleh faham? Okay, so now we are going to study in detail. Okay, cikgu nak pergi kepada ni. Okay, kita pergi kepada isotonic dulu. Okay, awak tengok ni, konsep isotonic. Apa maksud isotonic solution? Okay, isotonic solution is produced when solution A and solution B have the same concentration of solution. Okay, so awak tengok this is selective permeable membrane. So this will represent plasma membrane. Yang ni takkanlah awak nak ambil plasma membrane pada sel. So kita kena ada something yang menyerupai ciri-ciri plasma membrane. So kita guna apa? Kita guna selective permeable membrane. Okay. Kita guna ni sebagai to represent plasma membrane. Okay. Okay, so this is region A. This is region B. Okay, so kita pergi satu-satu. Awak tengok A ni. A ni, okay. We have to recognize, okay, the molecule first. Okay, so yang bulat-bulat biru ni, okay, this is water molecule. Okay, yang bulat besar warna merah ni, ini adalah solute. Solute dalam bahasa Melayu, bahan pelarut. Okay, bahan solute ni, the example could be sugar, salt. Okay, macam dia larut kan, sebagai contoh gula kan. Bila awak tak nak air, dia larut tak? Larut lah. So larut tu kita namakan sebagai solute. Okay, so kita tak general saja. It could be salt, it could be sugar. So kita name it as a solute. So how many solute you have in A and how many solute you have in B? So A you have 2. So B also you have 2. So right now you can say that A and B they have the same concentration of solution. Why? Because the number of solute in A and the number of solute in B are the same. Dua-dua. Boleh faham? Sebab tu kita cakap dia adalah isotonic solution. Why? Because they have the same concentration of solution. Explanation. Solution A and B are isotonic towards each other. So, no net movement of water. So, kalau awak tengok this show to you the direction of water. So, for example, okay, remember, even though they have the same concentration, the diffusion of water still occur. Okay, masih lagi berlaku. Tapi, the amount is the same. Sebagai contoh, okay, awak kena ingat since it has selective permeable membrane, it will only allow small substance to pass through it. Maksudnya, hanya water molecule saja yang boleh bergerak. Solute ni tak boleh bergerak, dia akan remain kat tempat dia. Okay, so direction yang awak tengok error ni kiri ke kanan ni, ini menunjukkan diffusion of water. Okay, so for example, when two molecule of water diffuse from A to B, okay, and then another two molecule of water will diffuse from B to A. Sebab tu tak ada no net movement of water. Sebab apa? Dua pergi daripada A, dua pergi daripada B. So dua tolak dua, berapa? Kosong. Sebab tu no net of water movement. Okay. Okay, the next one, hypotonic. Awak ingat hypotonic ni? Hypotonic ni maksudnya dia banyak air. Dia ada banyak air berbanding solute. Dia ada banyak air berbanding solute. Okay, so kita tengok So sekarang ni, how can you compare which one has higher water concentration? Okay, so kalau kita tengok kat sini, A dengan B. A ni ada berapa solute? Ada satu. B ada berapa? 1, 2, 3. Dia ada 3 solute. So A dengan B ni siapa yang lagi banyak molekul air? Mestilah A kan? Sebab apa A ada banyak water molecule? Sebab dia ada kurang solute. Maksudnya dia cair. Yang ni pekat. Sebab apa pekat? Sebab dia ada banyak solute. So sebab tu dia cakap solution A has a low solute concentration and higher water potential ataupun higher water concentration. So I would say solution A is hypotonic to the solution B. Okay, so kita boleh cakap solution A ni adalah hypotonic berbanding solution B. So what is the result? More water diffuse from solution A to solution B by osmosis. Awak tengok pergerakan air. Pergerakan air ni satu sahaja, iaitu daripada A ke B. Sebab apa? Air lebih banyak berada di sini berbanding di sini. So air ni dia akan bergerak daripada tempat banyak ke tempat sedikit. Tempat banyak apa? Banyak air lah maksudnya. Okay. So at the end of the experiment, you can see the level of water in A would be lower. Dia akan jadi kecil, jadi sikit sini jadi banyak. Sebab apa? More water from A diffuse into B by osmosis. Tapi kalau yang ni awak tengok sama kan? Dua pergi, dua masuk. So tak ada apa berbeza lah. So in the end of the experiment, if you have isotonic solution, okay, the level of water in A and B are equal. Sama. Tapi kalau hypotonic solution, A akan jadi kurang. Sebab apa? Banyak air bergerak daripada A kepada B. Okay, and the last one is hypertonic. Kalau hypo tadi, awak belajar, hypo ni, okay, dia ada banyak water potential. Tapi kalau hyper, awak tukar je lah. Okay, dia ada higher water solute. Macam hypo, dia ada banyak air. Kalau hyper ada banyak solute macam garam ataupun gula. Maksudnya dia kurang air lah. Apa maksud hypertonic solution? A solution that has higher solute concentration. Terbalik. Macam ini low, yang ni high. But low water potential. Dia ada kurang air. So kita A dengan B awak tengok. Okay, kalau A ni ada berapa? Ada berapa solute? 1, 2, 3. B ada berapa? B ada 1 sahaja. B ada 1 sahaja. So kita cakap A ni adalah hypertonic berbanding B. So what will happen? Okay, ingat solid tak bergerak. Hanya yang bergerak hanyalah water molecule. So more water from B will diffuse to the A. Dia sebenarnya konsepnya sama macam yang atas ni. Awak tengok. Yang ni dia bergerak daripada A ke B. Yang ni bergerak daripada B ke A. Sebab apa? A ni adalah hypotonic solution. Kalau yang ni B lah hypotonic. Air akan bergerak daripada hypo ke hyper. Yang pada banyak air pergi ke kurang air. Boleh faham? So A is a hypertonic solution to B. So more water diffused from B to A by osmosis. So at the end of the experiment, you will see the level of water in A is higher than in the B. Okay. Okay, if you don't understand, you can rewatch this video and make some note. Okay. Okay guys, so let's continue. Okay, yang ni this is the general information about iso, hypo dengan hypertonic solution. Okay, cikgu nak tambah satu lagi ya. Okay, what is the example of isotonic solution, hypotonic solution and hypertonic solution? Okay lah, nak senang cerita, hypotonic solution ni adalah solution yang ada banyak air. Banyak air tak ada langsung ataupun ada sedikit sahaja. Ada sedikit sahaja solute. For example, hypotonic solution ni contohnya adalah air kosong ataupun distilled water. Sebab apa distilled water? Sebab dalam distilled water ni hanya ada air semata-mata. So dia ada higher water potential ataupun higher water concentration. Kalau hypertonic apa cikgu? Contohnya air sirap yang ada gula. Air sirap ada gula kan? Mestilah ada banyak gula, ada sirap. So dia akan jadi pekat. Bila dia pekat, kita boleh cakap dia ada higher concentration of solute tapi ada low water potential. Tapi isotonic ni dia sama dengan sebagai contoh air apa isotonic drink. Yang awak minum tu apa contoh dia? 7 plus apa 100 plus 7 plus pula aku ni. Okay, 100 plus for example. Maksudnya concentration of water and glucose in isotonic drink with our body are equal. So bila awak minum, it won't affect anything. Dia akan awak hilang okay. Okay, next is about apa efek kalau kita guna hypotonic, hyper dan isotonic kepada animal and plant cell. Kita nak tengok apa efek apa efek solution yang tiga ni kepada animal dan juga plant cell. Okay, so we go for the animal cell dulu. Okay, we go for animal cell first. Okay, jap. Kita betulkan dulu. Okay, so the effect of hypo, iso, and hypertonic on animal cell. Okay guys, so usually in biology, animal cell ni, we use red blood cell. Kita guna sel darah merah. Usually we will use this kind of cell, to study the effect of different solution on animal cell. So jom kita tengok satu-satu ya. Awak tengok the effect of hypotonic solution. Hypotonic solution ni solution yang banyak air. Ada banyak air. So apa yang akan jadi? Awak tengok ya. When the red blood cell are placed in a hypotonic solution, water will diffuse into the cell by osmosis. Ini adalah red blood cell. So bila kita letak red blood cell dalam hypotonic solution, apa yang akan terjadi adalah air akan bergerak masuk ke dalam red blood cell. Banyak air akan masuk. Banyak air akan masuk. So by apa? By osmosis, causing the cell to swell and finally burst. So bila air banyak sangat masuk ni, sel ni akan mengembang. Swell tu mengembang ataupun membengkak. Dan finally burst. Burst tu pecah. Sebab tu awak tengok ni. Dia keluar ni. Burst. Dia pecah. This is because plasma membrane is too thin to withstand the osmotic pressure built in the cell. Sebab apa? Bila banyak air kat sini, osmotic pressure akan tinggi. And then, they also has a thin plasma membrane to support. And satu lagi sebenarnya adalah sebab red blood cell ni, it has no cell wall. Cell wall kan fungsi dia to maintain the shape of the cell. When the red blood cell ataupun animal cell has no cell wall, it means that it can maintain its shape. So that's why it finally bursts. Okay, the burst of red blood cell is known as a hemolysis. Okay, so bila sel tu burst, dia pecah, okay, kita namakan dia sebagai hemolysis. Okay. Okay, yang tadi kita pasal hypo. Sekarang ni hyper. Tadi kalau ingat hypo ni air masuk. Kalau hyper air keluar. Air keluar ni. Tengok macam durian pula lah. Okay, when red blood cell are placed in hypertonic solution, water will move out of the cell by osmosis. Tadi air masuk ni air keluar. Maksudnya when we put the red blood cell in the hypertonic solution, air yang apa maksudnya solution yang kurang air ataupun solution yang banyak solute. Yang pekat lah, pekat dengan garam, pekat dengan gula dan sebagainya. So apa yang akan jadi adalah more water will diffuse out. Air bakal banyak keluar. Okay, this will cause the cell to shrink. Dia akan mengecut. Dia akan mengecut. And the red blood cell are said to undergo crenation. Crenation ni adalah suatu proses yang mana banyak sangat air keluar daripada red blood cell. So that process is called crenation. Okay. Okay, and the last one, isotonic solution. Ingat isotonic solution is a solution that has the same concentration of water and solute with our cell. Okay, awak tengok. The water diffuse into and out of the cell by osmosis at the same rate. Maksudnya air masuk dan air keluar adalah sama. Sebab tu no net movement of water across plasma membrane. Okay, so maksudnya kalau contohlah bila kita letak red blood cell ni dalam isotonic solution sebagai contoh, okay, when 2 molecule water diffuse in, another 2 water molecule will diffuse out. So 2 masuk, 2 keluar. So 2 tolak 2 berapa? Kosong. Sebab tu no net movement of water. And this will make the cell maintain its normal shape. Sebab itulah red blood cell will not be affected. It will remain the same if you put in isotonic solution. Boleh faham? Okay guys, so that's all for animal cell. Okay, let's go for the plant cell. Kita tengok plant cell pula. Okay. Okay, the next one is plant cell. Awak tengok So, kita pergi kepada hypotonic solution dahulu. Hypotonic solution is a solution that has higher water potential. Ada banyak air. Okay. So, when plant cell are placed in the hypotonic solution, okay, sekejap. Sebelum tu cikgu nak cerita sikit lah. Okay, so usually in the lab, okay, untuk animal cell kita guna red blood cell. Okay, kalau untuk plant cell, we basically will use either kita akan guna potato ataupun kita akan guna onion. Kita guna kentang ataupun bawang to represent plant cell. Okay, just want to share with you. Okay, let's go. Tengok ni hypotonic solution. Apa jadi? When the plant cell are placed in hypotonic solution, more water will diffuse into the vacuole by osmosis. Okay, kita tahu this is plant cell. Dalam plant cell ada vacuole. Vacuole tu apa cikgu? Vacuole tu yang besar tu, awak tak ingat dah macam mana structure of animal cell. Okay, so air tu bila dia masuk ke dalam plant cell, dia akan masuk ke dalam vacuole dahulu. Sebab apa? Vacuole is the place where the water will be stored. Di mana air akan disimpan. Okay, this will cause the vacuole to expand. Bila banyak air masuk, mestilah vacuole tu mengembang. And push the cytoplasm and plasma membrane against the cell wall. Maksudnya, bila vacuole tu membesar, okay, vacuole tu ada higher osmotic pressure, betul tak? Bila ada banyak air, mestilah ada banyak osmotic pressure. So, osmotic pressure itulah yang akan menekan cytoplasm dan juga plasma membrane against cell wall. Kan kat ujung kat luar. Okay, contoh ini adalah plant cell. Ujung dia adalah cell wall. So bila vakuum yang besar tu tolak air, itu akan menyebabkan condition. Okay, the cell are said to be terjit. Terjit dalam bahasa Melayu segah. Dia akan jadi sergah. So in the exam, if they ask you, okay, what is the condition when the cell become turgid? So this is the answer. The vacuole expand and push the cytoplasm and plasma membrane against cell wall. Okay, tengok The plant cell does not burst because the cell wall is rigid and strong. Tapi kalau animal cell it burst kan bila air banyak masuk. Why? Because it has no cell wall to maintain the shape of the cell. But in the plant cell there is a cell wall. So that's why the plant cell does not burst. Okay, Tugger pressure is important to the plant cell because it gives support to maintain the shape of the cell. Tugger pressure ni is great when the vacuole has so many water. Bila dia ada so many water, it will push. Okay, it will push the cytoplasm and also it will push the plasma membrane. So this create a pressure and that pressure is called Tugger pressure. Why it is important? To give support and maintain the shape of the cell. Nampak segar lah. Kan? Air sayur kan bila ada banyak air kan dia akan segah lah kan. So cell turgidity cause the gut cell to swell and allow stomata to remain open for photosynthesis. So maksudnya bila dia turgid, bila dia turgid kita namakan sebagai cell turgidity lah. So apa yang bagus? So bila dia ada cell turgidity, stomata akan terbuka. So senang dia nak buat proses fotosintesis. Sebab apa? Kalau pokok yang layu sebagai contoh. Pokok yang layu maksudnya dia kurang air kan. Bila dia kurang air pun tak dia buat fotosintesis. Tak boleh. Tapi kalau pokok yang banyak air, dia boleh buat fotosintesis. Tak boleh. Sebab apa? Sebab dia boleh buka stomata dia. Stomata tu apa cikgu? Ini awak kena ingat balik lah. Stomata tu adalah sejenis lubang yang membenarkan oksigen masuk dan oksigen keluar. Okay guys, so that's all for hypotonic solution. The next one is hypertonic solution. Hypertonic solution is a solution that has higher concentration of solute. So what happened? You see here. Okay, when the plant cell are placed in a hypertonic solution, the water will diffuse out of the vacuole by osmosis. Ingat the water is stored in the vacuole. That's why water diffuse out from vacuole. Macam tadi water diffuse in, ni water diffuse out from where? From the vacuole by osmosis. So what will happen? The vacuole and cytoplasm will shrink. Dia akan mengecut. Ialah sebab keluar air kan. Causing the plasma membrane to be pulled off away from the cell wall. Okay. And this process is called plasmolysis. Plasmolysis will cause the leaf and stem to bend downwards and wilt. Dia akan jadi layu. Okay, plasmolyzed cell can regain their turgidity if the cell are returned to a hypotonic solution immediately. So if you see the plant cell become plasmolyzed, so one of the solution is that you put them in a hypotonic solution. Why hypotonic solution? Because hypotonic solution is solution that has higher water potential. Ada banyak air, so dia akan serap. And the cell are said to be deplasmolysis. Tadi dia plasmolysis kan. So bila kita letak air tu dalam hypotonic solution dan air banyak masuk, kita boleh cakap sel tu mengalami deplasmolysis. Okay, sebab apa? Banyak air akan masuk balik ke dalam sel yang kurang air tadi. And the last one is isotonic solution. Yang ni kita boleh cakap concentration of water and solute in the cell are equal. Okay, so what happened? Okay, when the cell sap of the plant cell and extracellular solution are isotonic, water potential is the same. Water potential sama maksud dengan water concentration. So the movement of water diffuse in and out is the same. And the cell become flaccid. Flaccid tu maksudnya dia tak berubah lah, dia sama sahaja. Okay, flaccid. Sebab apa flaccid? Sebab kita letak dalam air isotonik. Dan tidak ada perubahan. And then kita namakan dia sebagai sel yang mengalami flaccid. Yang mana flaccid tu is a condition when there is no different happen when we put them in the isotonic solution. Okay guys. Okay guys, so let's continue a little bit. Okay guys, so we just stop here. Okay, for this experiment, I think I want to teach you when you are already in the school, okay? Okay, let's go to the last subtopic for this chapter. Okay, the movement of substance across plasma membrane and its application in our daily life. Okay, so here you see, okay, kita ni tak ada apa pun. Kita tengok yang bawah ni. Phenomenon of water wilting. Okay, yang pokok layu awak tengok ni. This is a fresh plant. This is a wilted plant. Okay, the excessive use of fertilizer may cause wilting in the plant. So this is fresh plant. If you use excessive fertilizer, okay, dia akan menyebabkan pokok tu layu. Sebab apa? Ni awak tengok ni. Dissolved fertilizer will cause the soil water become hypertonic to the cell sap of the root. So bila kita guna banyak baja, okay, bila kita guna banyak baja, kita akan menyebabkan persekitaran dia, persekitaran dekat pokok tu menjadi hypertonic. So hypertonic apa jadi? So water will diffuse out by osmosis from the root cell, okay, to the soil. So bila air banyak keluar daripada root cell, okay, cell will become plasmolysed. Dia akan kurang air. And the cell in the plant will recover once they are watered. So cara nak mengatasinya adalah awak kena siram dia lah dengan air. Sebab apa dia kurang air? Kurang air sebab apa? Sebab dia ada hypertonic solution. Disebabkan awak guna banyak fertilizer. However, if the period of plasmolysis is prolonged, the wilted plant will eventually die. Tapi kalau awak tak treat dia dengan air, okay, dia akan matilah. Okay, the next one. Okay, the concept of movement of substance across plasma membrane has many applications in our daily life. For example, tengok, dehydration drinks such as oral rehydration salt will help to recover the loss of water and electrolyte in individual with diarrhea. Okay, diarrhea ni adalah suatu penyakit yang mana awak cerek-berek. Bila awak cerek-berek, awak mesti kurang air, betul tak? So that's why the doctor will give you ni oral rehydration salt. So apa? It will recover the water loss. Dia akan make sure awak punya air dalam badan okey. Lepas tu ni, diminum air apa ni? Isotonic drink. Ni isotonic drink will help athlete to recover the loss of water and electrolyte as a potassium and sodium through perspiration. Okay. So sebab itulah, okay, athlete they basically will consume, okay, isotonic drink. Okay. Why? To recover the water loss. Okay, the next one, saline solution. Ni saline. Okay, saline solution ni nama lain bagi larutan garam. Okay, so saline solution normally used in the medicine, okay, are isotonic to the blood plasma. So it contain, ni concentration dia ni. Maksudnya this is the concentration of sodium chloride. Sodium chloride ni garam. Okay, yang mana dia mempunyai kepekatan yang sama seperti darah kita. Okay, yang ni banyak jumpa kat hospital. And the last one, ni apa tengok. Liposome vesicle that contain aqueous solution surrounded by phospholipid bilayer membrane. Okay, liposome are used to protect drugs and active substance taken orally from being destroyed by gastric juice. This way, the drug can react to the target cell. Okay, so basically these are the things that normally use. Okay, the application of movement of substance. Okay. Okay, I think that's all. Oh, there's one more thing here. Okay. Okay, reverse osmosis is a technology commonly used to extract fresh water from seawater using desalination process. A reverse osmosis equipment, okay, a pressure is applied to push the seawater through semi-permeable membrane. Okay, the membrane will allow water molecule to pass through it, but not foreign subparticles, salt, and microorganisms. As a result, only pure water is released. Okay, so this is the thing that if you want to use the water from seawater. Okay, air laut ni usually, ni awak tengok air laut ni. Air laut ni dia ada banyak foreign particle, microorganism, salt and so on. So we use the application of osmosis here, where we use selective permeable membrane, where we only allow small substance, which is water molecule, to travel from this area to this area. So as a result, we only have water molecule here. So this is fresh water that can be safely used afterwards. Okay. Okay, guys, so I think that's all for today. So if you have any question, you can ask me. Please write down your name and class, and one student has to ask one question. Okay, please write down your question and name and class in the comment section. If you have any question, okay, please PM me personally. That's all for today. I see you next time. Okay, assalamualaikum. Bye bye.